Up next, we have Neelakshi Mungra from the University of Cape Town in South Africa, presenting the development of novel auristatin F-containing immunoconjugates for the treatment of triple-negative breast cancer. Neelakshi, whenever you're ready, take it away.
So my PhD project entails the development of SNAPTag-based fusion proteins, which will not only be used as diagnostic tools, but for triple-negative breast cancer, but also in the development of auristatin F immunoconjugates and photoimmunotheranostics. So today I will focus mainly on the generation of these auristatin F recombinant antibody-drug conjugates. Despite the surge in technological progress, breast cancer remains a major public health concern worldwide. It is a major cause of, it is a second leading cause of death in women around the world and the #1 cancer diagnosed among South African women, therefore making it an important health priority. The key to effective management of breast cancer lies in early and accurate diagnosis. In the clinical setting, breast cancer screening is typically done through a physical examination and through affordable imaging techniques such as X-ray mammography, magnetic resonance imaging, and ultrasound. However, these methods are not always accurate or effective. Biopsies are also taken and immunohistochemistry is used to identify tumor antigens in cells of a tissue section through the use of specific antibodies. This principle has thus allowed the histological classification of breast cancer. 4 intrinsic subtypes have been identified, namely luminal A, luminal B, HER2 positive, and basal-like subtypes, with basal-like having the worst prognosis. The majority of triple-negative breast cancers are of the basal-like subtype, and they lack expression of the estrogen receptor, the progesterone receptor, as well as the human epidermal growth factor 2 receptors. As such, triple-negative breast cancer account for 10 to 15% of all breast cancers, and they occur more predominantly in women of African ancestry. While this racial preference cannot be explained, triple-negative breast cancers represent the most aggressive and heterogeneous subtype, and up to now, no targeted therapy is currently available, which increases the dependency on conventional treatments. If we have a look at the treatment approaches available for breast cancer, we find that they can be broadly classified into non-targeted and targeted therapies. Non-targeted treatments such as chemotherapy, surgery, radiation and photodynamic therapies are associated with several side effects and limitations, spurring the focus towards a more targeted approach, often using antibodies. In this project, we will focus primarily on antibody-drug conjugates. So what are antibody-drug conjugates? These consist of a monoclonal antibody, which is linked to a toxic payload, often an antimitotic or a DNA disrupting agent, via a cleavable or a non-cleavable linker. They were designed to improve the efficacy and reduce the toxicity of highly potent drugs, which are too toxic on their own to be systemically administered. They work by binding to the cell surface and to the cell surface antigen on the surface of a tumor cell, and this is followed by the internalization. And we have lysosomal degradation of the protein component, which releases the active payload into the cytosol, where it can interact with DNA or with microtubules in order to arrest the cell cycle and cause apoptotic cell death. Despite over 3 decades of research, only 7 antibody-drug conjugates have been approved by the FDA, which highlights challenges in their development. Several aspects of the antibody, the linker, and the toxic payload must be taken into account, since they can affect the stability, the pharmacokinetics, and also give rise to side effects. For example, most conjugation methods available to all monoclonal antibodies with effector molecules rely on the direct functionalization of lysine side chains or the reduction of disulfide bonds on antibodies. However, these methods give rise to a heterogeneous mixture of products with different drug-to-antibody ratios, pharmacokinetic and toxicity profiles. A proposed solution involves the use of self-labeling proteins, which would allow the site-specific conjugation and the production of homogeneous conjugate preparations. SNAPTag is a mutated version of the human DNA repair enzyme, alkyl-guanine DNA alkyl transferase, which can be fused to virtually any protein of interest, in our case, single-chain antibody fragments, and it can react irreversibly with benzyl guanine modified substrate to generate a recombinant product. These substrates can be fluorescent dyes, photosensitizers, or cytotoxic drugs. At the MBNI research unit, we envisage to use these SNAPTag-based fusion proteins to develop a portfolio of companion immunodiagnostic tools that could eventually be used in multiplex immunofluorescence imaging of various biopsies. This approach would allow the determination and quantification of several target proteins as well as their location while preserving the tissue architecture. The benzyl guanine modified substrate that I propose to use in the study is benzyl guanine auristatin F. Auristatins are synthetic derivatives of dolastatin 10, which is derived from the sea hare Dolabella auricularia and found to have antimitotic properties. They inhibit cell division by blocking the polymerization of microtubules, and they are known for their high potency. Auristatin F was favored due to its lower membrane permeability and ability to be conjugated via non-cleavable linkers without the loss of cytotoxic activity. This project aims to exploit the differential expression of CSPG4 and CD44 in triple-negative breast cancer. Chondroitin sulfate protoglycan 4 is a transmembrane protein, which is highly conserved and expressed on 73% of triple-negative breast cancer lesions and cell lines. It is associated with survival, metastasis, and poor treatment outcomes, amongst others. On the other hand, CD44, a cancer stem cell marker, is a transmembrane glycoprotein which plays a major role in malignant formation, growth, invasion, metastasis, and recurrence. Therefore, the aims of this project are as follows: to generate SNAPTag-based fusion proteins, which will be used to screen triple-negative breast cancer cell lines for CSPG4 and CD44 expression. These same fusion proteins will then be used by Natasha to validate expression of these targets on South African breast cancer biopsies. The second aim will involve the generation of novel auristatin F-containing recombinant antibody-drug conjugates targeting CSPG4 and CD44, and to assess their cytotoxic activities in vitro. This is an overview of the methodology used, starting with the in silico design and molecular cloning of the expression constructs. We then transfect the plasmid DNA in HEC293 T cells for protein expression. After harvesting the cell culture supernatant, we purify the fusion protein using immobilized metal affinity chromatography, and we validate protein expression and integrity using SDS page and Western blot before assessing the binding potential of the antibody fragment as well as the cytotoxic potential of our antimitotic drug. Section 1 will look at the molecular cloning of these SNAPTag-based fusion proteins. Figure 1A in the top panel shows us the open reading frames coding for these proteins. SnapGene software was then used to introduce these open reading frames into mammalian expression plasmids. We then conduct the cloning in the lab, and after cloning, we transfect these plasmids into HEC293 T cells. Here, visualization of green fluorescent protein, as seen under the green channel, shows successful transfection as well as in cis expression of our putative fusion protein. Section 2, engineering of anti-CSPG4 scFv SNAP. A total of 1 liter volume of cell culture supernatant was purified. Figure A shows us the chromatogram after purification. The area under the curve gives us an estimation of the amount of protein being eluted over time. In figure B, an SDS page gel of the pooled fractions was run. The red arrow indicates presence of the fusion protein at around 54 kilodalton, which corresponds with the theoretical size. However, we can see the presence of higher bands in peaks 1 and 2, which suggests that the protein purity might be compromised. In an ideal setting, a second protein purification step, for example, size exclusion chromatography, would be needed after IMAC. In this case, we did densitometry measurements using ImageJ software in order to determine the percentage of scFv SNAP protein present in the total protein sample. These values were then used in downstream calculations. Next, the fusion protein was conjugated to benzyl guanine alexafluor 488, which is a fluorescent dye. This experiment confirms successful expression of the fusion protein as well as the functionality of the SNAPTag component. And this is shown by the fluorescent band observed at around 55 kilodalton. Thereafter, tumor cells were incubated with this Alexa 488 conjugated protein to assess binding qualitatively via confocal microscopy. From figures A and B, we can see binding of our anti-CSPG4 scFv SNAP protein to the surface of SKML28 cells and HS578T cells, which are indeed positive for CSPG4. Similarly, surface binding was observed on MDA-MB468 cells, but no binding was observed on MDA-MB231 cells. After confirming the functionality of the antibody moiety and SNAPTag, I then conjugated the fusion protein to benzyl guanine modified auristatin F. However, in order to confirm saturation of the fusion protein with the antimitotic drug, I then performed an additional conjugation, this time with benzyl guanine modified Alexa 488, which would allow visualization of the resulting compound. No fluorescent band was observed in lanes 1 and 2, despite presence of the protein, which confirms saturation with benzyl guanine auristatin F. An XTT-based cell viability was then conducted to determine the cytotoxic activity of the auristatin F immunoconjugate. As shown by the graphs, dose-dependent decrease in cell viability was observed on HS578T cells, MDA-MB468 cells, and SKML28 cells, but not on the HEC293T cells or MDA231 cells. The IC50 value gives us an indication of the cytotoxic potential. The lower the IC50 value, the better the cell killing ability. In this case, the IC50 value ranged from 190 nanomolar to 1.66 micromolar. Lastly, the same experiments were conducted with anti-CD44 scFv SNAPF. Of note, SNAPF is just a false labeling variant of SNAPTag, which should display higher reactivity towards benzyl guanine substrates. A total of 600 milliliter of cell culture supernatant was purified, and the concentrated protein fractions were then run on an SDS page gel. The red arrow shows presence of the protein at approximately 50 kilodalton, which corresponds with the theoretical size. A standard curve was then drawn, and the amount of protein was assessed by densitometry. Next, conjugation with benzyl guanine Alexa 488 at the required size was observed, confirming the functionality of the SNAPTag component. Confocal microscopy was then carried out, which confirmed surface binding to HS578T cells, MCF7 cells, and MDA-MB468 cells, but not to MDA231 cells. Conjugation with auristatin F revealed IC50 values ranging from 839.7 nanomolar in HS578T cells, 1.27 micromolar in MCF7 cells, and 100.4 nanomolar in MDA468 cells. In conclusion, given their observed binding data, the SNAPTag-based fusion proteins generated show potential as diagnostic agents for the establishment of multiplex immunofluorescence imaging. And this was also confirmed by my colleague Natasha, who was able to demonstrate for the first time, differential expression of CSPG4 and CD44 on South African triple-negative breast cancer biopsies. This study also demonstrates the proof of concept for the establishment of methodologies centered around the generation of novel recombinant and homogeneous ADCs targeting CSPG4 and CD44. According to the work of Wojciech et al., the IC50 values were ranging from 0.6 to 12 nanomolar. And in this study, we obtained 3-digit nanomolar to micromolar range. However, we cannot compare IC50 values of different ADCs across cell lines due to multiple varying parameters, such as the receptor expression, the internalization rate, the trafficking, the intracellular concentration of the payload, as well as elevated levels of drug efflux pumps. Therefore, for future work, flow cytometry will be used to assess the level of receptor status on tumor cells in order to inform the selection of the best expressing cell lines for further cell viability assays. The final take-home message is that with the establishment of a robust companion diagnostic tool provided by SnapTag, we aim to be able to screen and recognize patients which are most likely to benefit from the recombinant immunotherapeutics being currently generated. In consequence, by tailoring treatments that suit the individual patient, we will foster the concept of precision medicine, which will allow more effective management of a daunting disease like African triple-negative breast cancer. Last but not the least, I would like to thank and acknowledge my supervisor, Professor Stefan Boff, my colleagues at the MBNI Research Unit, as well as all our funders. Thank you all for your attention.
Thank you for that great presentation, Neelakshi. We just have one question for you in the chat.
Okay.
An attendee would like to know if you expect any different results regarding the other breast cancer subtypes.
Sorry, can you repeat the question?
Yeah, no problem. If you expect any, if you'd expect any different results regarding the other breast cancer subtypes.
Is the person referring to the cytotoxicity results?
I'm not too sure, they didn't clarify. If they want to message in the chat, you guys could message in the chat and clarify that.
I think the question might be if the findings are specific to triple-negative breast cancer, or if you envision, similarities in some other aggressive forms of cancers.
So I think it will depend on the cell type, because it all boils down to the level of receptor status on the tumor cell. That's what I've been finding. Like if I have a cell line that, for example, does not express a high level of CSPG4, which is my target antigen, then I would not see such a good dose-dependent killing of the cell population. So from this, we can then predict that in other cell lines, in other types or forms of cancers, we will see different effects. So it would be important to test these before trying to, for example, conduct in vivo studies.
I guess if I can just jump in with a quick question here. You mentioned you've done some preliminary studies in some biopsies. So what kind of distribution are you seeing of these? Is it pervasive throughout, for instance, in a breast cancer, or is it, you mentioned tissue specific? How does that look?
Okay, so I am not really the expert on the biopsy work, but what my colleague has found is that you would see a different distribution of these receptors based on, for example, the ethnicity of the patient, the age, other factors like if they have other comorbidities, stuff like that, and this would affect what you would see on, for example, how the architecture looks like. Yeah, I wish I had pictures for that, but probably next time. I think it would have been interesting to show. Yes.
I'd love to see them next time. Perfect. Thank you.
Yes. Thank you so much.
Okay, thank you, Neelakshi.
Thank you.